Hello, this lecture is entitled The Goal of Clinical Drug Development. To understand model-based drug development, you need to understand the final deliverables of clinical development. So when a company is running a series of clinical trials, what are they ultimately hoping to achieve? Well, clearly, the goal here is to develop a set of data and a set of results which clearly facilitates a risk-benefit assessment of the investigational drug. So that's both for the benefit of the pharmaceutical company, for the regulatory agencies, and ultimately for patients. With this in place, it will allow a sensible and judicious selection of the starting dose and the maximum dose which could be approved for use in the patient population. So by the end of phase 3, we would need an accurate and precise assessment of the primary efficacy endpoint. Here is an example shown where we can see that as we increase with dose, we increase with effect. And here the shaded area is a 95% prediction interval. So we think at 16 milligrams, for example, we think the effect is around 3.5, but it could be up as high as 4.8 or down as low as 2. Often we actually have multiple efficacy endpoints. So here we have again the same graph as we've seen before for our efficacy endpoint 1, but we also have an efficacy endpoint 2 down here. Now these curves could be quite similar or they could be quite dissimilar. And finally, we would have multiple safety endpoints as well. So here is 2 examples of a continuous endpoint. So here we have something like heart rate, and we can see that as we go across the dose range, there's an increase potentially in heart rate, perhaps to some concerning levels. And similarly down here we can have an adverse event like nausea or nausea and vomiting, where again as we go across the dose range we see an increase in effect. So what we might ask is the precision of this prediction interval here at this particular dose of 8 milligrams, is that adequate enough? So here we have a best guess that the drug only induces an effect of around 0.4 beats per minute. But we also see the upper prediction interval is 1.9. So wouldn't this be better? Now what we've done versus the previous slide is we've seen that there's a reduction in the uncertainty in this estimate now. So before we had about 1.9 as the upper estimate, and now it's 1.3. So you can achieve that by doubling the sample size, would achieve that same type of result, or by selecting your doses more cleverly, so improving the design of your phase 2 and phase 3 programs. So what's wrong with looking at a single dose level in phase 3? It's very common. Companies are interested in only looking at perhaps a single dose. Well, here is the same type of results now, but only looking at a single dose. So this would be 16 milligrams, for example, versus placebo. And we have an estimate of the effect for our 2 efficacy endpoints and an estimate for our safety endpoints as well. Now the concern here is that we really don't know how to position these results. So higher doses could have a higher utility. By utility, we mean they have a better benefit-risk profile. So here, as you increase the dose, perhaps you're going to get a very large gain in efficacy relative to only a modest worsening in the safety profile. Or perhaps the lower doses would have a higher utility, so they would have a better risk-benefit. So if we go to lower doses here, we're going to get a small reduction in the efficacy, but a large improvement in the safety. So really having a single dose here is hopeless for rational decision making. It doesn't allow you the opportunity to position your drug dose level. Using model-based drug development, we can also think about looking at the differences versus a reference dose. So here, for example, we've plotted the estimate and the 95% prediction interval versus 16 milligram. So obviously 16 milligram versus 16 milligram is exactly the same, but the lower doses we're losing now on efficacy and the higher doses we're gaining. And similarly over here from a safety perspective, we see that we improve when we go to lower doses and we get worse as we go to higher doses. So it allows us to try to work out what would be a sensible starting dose, what would be a sensible final dose, perhaps the highest dose. So a brief summary of the goals of clinical drug development when we think within a model-based drug development philosophy it's important to describe the dose-response relationships for multiple efficacy and multiple safety endpoints, not just a single dose level. This allows the utility, so the benefit risk, to be determined across the dose range. This allows a rational selection of a low or starting dose and the rational selection of a maximum dose, allowing the physicians to have a range of doses that they can reasonably prescribe. Important in this process is the precision of the predicted dose responses. So when we talk about precision, we're talking about how narrow or wide those 95% prediction intervals are. Clearly we want to control that by running studies that are sufficiently large that we get a good estimate of the effects of the drug. So when we think about the integrated model-based drug development analysis, when we're designing the phase 2 studies and the phase 3 studies, we need to be thinking about putting all that data together to construct the types of plots that we've shown in this short presentation. And then you can make some rational decisions on drug development on the basis of that.
I hope you found this video interesting. If so, you may want to check out our other videos. If you have any comments or suggestions, please feel free to contact me.